Our next speaker is Joanne from Gordian Biotechnology.
Good morning, everyone. As you can tell from the length of my title, when I submitted this abstract, I was fully intending this to be a poster, so I'm thrilled to be up here, and I thank the organizers for giving an opportunity to present our unique screening platform. Before I begin, I just want to say I'm the messenger. Here are the people who did the actual work, including our CEO, Francisco Laporte here. And he is at the conference with me, and he's an absolute prodigy at handling very, very large amounts of data. And also here with me is Jingshu Chen, who's the Metabolism Program's single cell scientist. I'd also like to thank Scott Friedman, who's been quick with his email responses and generous with his time, and I couldn't ask for a better scientific advisory board member. So human population genetics have yielded a cornucopia of targets for drugs, but unless you're lucky enough that the locus co-localizes with an eQTL in your tissue of interest, or is in strong linkage disequilibrium with one, or has a exonic variant that, obviously damaging, you don't know whether we should be inhibiting or activating the genetic target. And in the case of FTO here, it might not even be the gene or any gene within the vicinity. So you have your work cut out for you if you take the traditional approach, simply knocking down or overexpressing just one gene at each of these loci, yields 106 interventions. If you take a small group of n equals 6 mice per group, this is over 600 mice and countless numbers of postdoc and scientist hours. At Gordian, we query the equivalent of this many interventions, plus basically it could be a few genes around each of these genes in these 53 loci in the equivalent of, 7 mice worth of liver. And how we do this is that we inject a pool of interventions that are identified by unique barcode. They're packaged into AAV8 for the liver program and is delivered into a MASH model. Now, because we need so few animals, we can afford to use models that take a lot of time and expense to generate. The GAN mouse was recently identified by the Litmus Consortium to have the highest human proximity score, and we feed them to 62 weeks to permit us to assess the HCC risk of an intervention. We also have a colony of African green monkeys done in collaboration with Wake Forest University. These monkeys were fed a high-fat, high-cholesterol, and fructose diet for 2 years. This is not a typo. And they developed portal fibrosis, which has a stay out of sight data suggests is more predictive of outcomes. The dose of AAV is titrated carefully so that only one intervention is transduced. Each hepatocyte is transduced by only one intervention, and transduced hepatocytes are rarely touching each other. So this enables us to isolate the effect of an intervention. We analyze these hepatocytes by single nucleus RNA sequencing, and whereas we do measure the expression of every single expressed gene in these hepatocytes, we like to bunch them into features to facilitate the analysis. And what this ends up looking like is shown here. So as you see, when we overexpress IL-6 in these hepatocytes, it is the strongest inducer of IL-6 signaling, along with a raft of other deleterious disease features. The Sense-Creviroc, I think I'm pronouncing that correctly, was shown to be outperformed by placebo in the F2 patients in the clinical trial, and our disease feature analysis would have predicted this. As you see here, it increased the STAHEP signature derived by Rodrigo Carlesi. Similarly, So then we did this by expressing an anti-CCL2 construct to mimic the effect of Syncreviroc. Similarly, dominant negative TLR4 construct increases signal in this deleterious feature. And if you recall from years ago, there was a Japanese company that had a TLR4 antagonist that was also outperformed by placebo. So this disease feature analysis would have predicted this. STING1 increases the NLRP3 inflammasome strongly. So again, showing how well this platform works. And we can curate features to query a number or any pathway imaginable. And one thing we're interested in doing is looking at hepatocyte regeneration. Admittedly, there are actually other companies doing in vivo screening in the liver to identify regeneration targets. But by simply just counting the number of hepatocytes that contain an intervention, you can't distinguish the fine line between restorative hepatocyte regeneration and tumorigenesis. But we can, and we can do so by looking for targets that may upregulate, say, the beneficial p53 pathway and Wnt signaling to bring about cell proliferation, while avoiding perhaps the more pathological YAP-TAZ pathway. So let's apply this to some target genes identified in GWAS. As shown here, TM6SF2 overexpression strongly decreases de novo lipogenesis according to this analysis, and this was shown in mice when that overexpressed TM6SF2. Likewise, PPP1R3B decreases DNL, and we know that this variant is associated with glycogen deposition, not fat deposition. That accounts for the liver enzyme rise. MBOAT7, a variant that with reduced activity has been long known to be associated with MASH, and overexpressing it here decreases activity of the NLRP3 inflammasome. And if we look at the other end of the spectrum, we see that mTARC1 overexpression really raises the activity of this disease feature, and we know that truncation variants are strongly protective against cirrhosis. So what I just showed you was just a little snippet of what we look at when selecting hits to proceed with. So this is an example screen here, and we look for interventions with a possibly beneficial combo of a variety of features, and we selected these for in vivo physiological validation. Here we have, we went back to the GAN diet mice, housed at thermal neutrality, and we timed the diet so that they are at F2 when we start the experiment, and there is tumor burden at the end of the experiment, so we can, again, mitigate the HCC risk. The mice are given a single saturating therapy, encoding the candidate intervention, and because these livers are so diseased, there's a lot of hepatocyte turnover, which necessitates a second dose. But right before the second dose, even when the expression is at a nadir, we see that in 6 of the 10 interventions that we queried, there was a significant reduction in ALT, and when looking at target 3 here, we see a sustained reduction in a variety of serum biomarkers of fibrosis. The bulk RNA-seq data corroborates this observation. Target 3 overexpression reduces fibrosis based on ECM gene expression in the curated gene set and unbiased gene set enrichment analysis. And there's also indication that target 3 would reduce inflammation. What I've shown you so far was overexpression screening. We can also do knockdown, and as you see here, knocking down ABCB11, which is cholestasis gene, puts the intervention at this end of the spectrum of the heat map. ABCB4, another cholestasis gene, clusters closely. it was very gratifying, therefore, to see that target 3 clusters at this end, along with knockdown of thyroid hormone receptor beta. Knockdown of SCAP might have a therapeutic effect since it's over at the other end here. And we also have a number of interventions that might be showing a positive effect. And, I'm sure many of you know here that it's kind of a bummer to spend years on a program and have it killed in the end by an NHP study because of a liver tox signal. Because of our screening platform, we can de-risk this upfront. So the left lobe of the vervet liver is the equivalent of about 5 mice worth of liver. So we can query quite a few interventions without killing the monkey, thereby alleviating any ethical concerns of using NHPs for early discovery work. And overexpressing PNPLA3 here seems to have a deleterious effect as expected. Overexpressing ADH1B, which is a primate-specific alcohol dehydrogenase, seems to have a beneficial effect in this system. So these monkeys are given a diet of marshmallows and white chocolate chip wafers, but not bourbon. So I was quite surprised to see such a strong beneficial effect of ADH1B here. But Nagochalasani's paper in 2020 in gastroenterology suggests that the ADH1B STAR2 variant does have a beneficial effect independent of alcohol consumption. And last data slide, we've adapted this to adipose tissue. We've successfully transduced multiple adipose depots in both mice and rats. And I'm in talks with Wake Forest, and it seems like we can also do this in monkeys without, again, killing the monkey. So proof of concept screening data should be available in just over a month, so stay tuned. And I'd just like to leave you with these key takeaways that we could curate these disease features to really query any liver disease, including decompensated cirrhosis, and we're showing promising targets in the lockdown screening, and we can do this in adipose. And hopefully, we can leverage this platform to examine anti-obesity agents to make sure there isn't any ectopic pathological deposition in the liver instead of the adipose. And I urge you to stop by the accompanying poster tonight, which we examine whether we could look at the hepatokine signature to find anti-obesity agents. I know it's unlikely that one hepatokine is a silver bullet, but what if we can reprogram the liver to secrete a nice suite of the beneficial ones? Thank you.
We have time for some questions.
Mahmoud Hussain from NYU Long Island School of Medicine. So you are transducing monkeys with AAV. How long does this effect last?
How long does the
AAV last in the liver? And you say you don't need to kill the monkeys, but if they have been transduced with AAV, can you do any other experiments in those monkeys? Is this useful?
Well, we know of actually multiple serotypes that can transduce the monkey. So in theory, we can transduce it with multiple libraries if we want to. We haven't done that yet. So the work from UPenn, such as Jenny Gregg, shows that monkeys do mount quite an immune response to the AAV. So we have to titrate the dose to minimize this effect. And then there's a peak of expression around 2 weeks, and then it kind of stabilizes at a low level over time. And so we've timed it. So the 4 to 6 week time point we've discovered is kind of like the sweet spot.
So you transduce and then do your analysis after 4 to 6 weeks?
Yeah.
And I saw that you did 2 transductions in mice.
Mm-hmm.
What was the lethality in those mice? Because there should be an immune response to second.
Actually, no. The mortality was, if anything, just from being on the GAN diet for over a year. There was no excessive mortality that we could observe.
Thank you.
Hi, Joanne. Very nice. You were talking about the titration so that you get individual cells that get infected. But how many cells, when you collect these and look for the barcodes, how many cells are infected with the same type. So I'm assuming it's not just one cell for one gene target.
Oh yeah, no, we certainly need more than that. So we're starting off with about 1,000 cells per intervention. So we have to collect quite a bit of data, but we're improving our technology and we're constantly bringing down this number. We're like increasing the signal to noise ratio. So yeah, certainly.
And have you played around, and this is probably more of a rodent experiment because of the sensitivity around NHPs, can you eventually get to where you might be able to look for multi-targeting? So you're looking for 2 or 3 hits in a single cell so that you can actually look what that combined effect might be.
Definitely. That's something we want to eventually get to so we can query combo therapies. Yeah.
Would the titration though kind of bias your screens to miss like quorum sort of effects where cells next to each other talk to each other?
Yes, definitely. But what I've found is that these second order effects, because they recruit immune cells, there's a lot of crosstalk between this and them and say stellate cells. These second order transcriptomic effects have actually proven to be very informative.
Awesome. Thanks a lot, Joanne.